Dr. Michal Kowalewski. Michael is the John L. and A. Beverly Thompson Chair of Invertebrate Paleontology, curator and professor in the Florida Museum of Natural History at the University of Florida. He also maintains affiliate appointments as a professor in the departments of geology, biology, and the School of Natural Resources and Environment. And prior to arriving at the University of Florida, and I don't know what I ate. I ate lunch with Michael and my stomach's been grumbling. Anyways, prior to arriving at the University of Florida, Michael was a professor in the Department of Geosciences at Virginia Tech. He's a fellow of the Geological Society of America, Institute of Advanced Studies at the University of Bologna, and the Paleontological Society. I mean, he's received numerous other awards and honors during his career. Suffice it to say, the list is long, right? And I don't want to cut into his seminar time, so I'll forego that. But his interests are quite broad, and they range from paleoecology, toponymy, geochronology, stratigraphy, quantitative methods, marine ecology, and conservation biology. And it's the latter that actually introduced me to Michael. And as I said before, I've had the pleasure of working with him but on a number of projects. We've co-advised or supervised a number of students together. He's an exceptional scientist, a great mentor, and a valued colleague. So we're fortunate to have him today as our speaker. So thanks, Michael, for taking the time out of your very busy schedule to wow us with all the cool things.
We'll see about that. Thank you, Tom. Such a thrill to do real presentations, after COVID, and we still don't do this often enough, and so I'm really excited to do this, and I'm very thankful that many of you decided to brave the weather and come here for my presentation. So it's also first time for me to be here in your college. And I had a great day. I met multiple people, and I had really fun conversations about science and other things. So I really appreciate being here, and I hope it's not my only time. And I'll be visiting again. And I promise I won't do more seminars. Clicking to new ones, good enough. So what I will try to present today is kind of an overview of 3 decades of research, and that's sort of all linked to this idea that historical data that can be provided by, in particular, the youngest fossil record, can actually provide some useful information that can benefit conservation efforts. And we're still a very young discipline. We are having a lot of challenges translating our science into some practical, actionable actions. But I hope we will eventually get there. And so a lot of it is still non-applied, but I hope to convince you that there is a potential there. And this is by no means a complete list, but because I'm sort of reviewing 30 years of research, as you may imagine, this is an effort by many different people, especially many students and postdocs over the years who helped us in various funding sources that cumulatively helped them. And many others is really sort of an understatement here. I would just notice here that John Hyman was a student at the University of Florida, now he's a postdoc here with Tom, so I was really thrilled to see him again today. So what I will try to do is kind of have 2 parts to my presentation. In the first part, maybe about 20-25 minutes, we'll just talk generally about what, at least to my understanding, what is conservation paleobiology and why all of you should be excited about it. And then following that, I will try to sort of showcase examples from our own research, some local, some not so local, to sort of bring home the generalizations that I will present in the first part. And I very much doubt I will be able to go through all 4 stories, so this is a menu, but I'm hopeful I can do at least 2 case studies, maybe 3, and we'll see how it goes. Partly depends on your patience. And I was trying to use the USF colors for the numbers. I'm not sure they came out okay. By the way, notice that I am the only person I know who correctly numbers slides. They should be descending, right? So there's 59 more. So it's completely useless when you number it like from 1 to 100. It doesn't tell the audience anything. So more seriously, the main reason why we may want to be interested in the geohistorical archives is simply the time dimension, and this is pretty obvious. When we look at what I would call contemporary scientific data, where we have real-time instrumentation, when we collect rigorous state bioinventorying data, really most of these data are from the last few decades. In many regions, it may be only a few years. And we have very few case examples of real-time data that go back to even 1950s. So essentially, we have virtually no rigorous scientific data prior to Industrial Revolution. And we have various types of data, though, that reach farther back in time, including written records, including early naturalists. But then we have also archaeological evidence, which is a huge source of information about how humans interface with nature. And then we have paleontological data. And these essentially can reach as far as we can. And so my argument is that they can really augment the neontological knowledge. So the contemporary scientific data are absolutely critical. We cannot do anything without those. But my argument is that we can enhance them further by looking at historical perspective. And this is a very weird plot, and I don't know if you noticed that the time scale is logarithmic actually, which, we never plot time on a log scale, but if I use linear scale, all these black bars will become little dashes, because essentially this is very, very tiny amount of time, not geologically speaking, but speaking from the perspective of human history on this planet, which of course reaches back thousands of years. And we can sort of make some very obvious and self-evident observations about why these data can help us to maybe make a better assessment and therefore maybe make better restoration or prepare better restoration targets. So for example, I sort of have this very simple and naive diagram showing a situation where if we are relying on modern scientific data exclusively, this is what we see. And we have some time depth, but maybe only decades. So for example, in terms of biodiversity, our scientific data may indicate that there are only two species in a clade, whereas if we actually extend this to geohistorical records, to fossil materials, we may actually discover that that clade was actually much more diverse before we started collecting modern data. And in fact, each of those examples can be backed up by examples from the literature. Like, for example, the highland birds that if we were to rely only on modern scientific data, we would think there are only two species in a clade, whereas in fact, once you add archaeological and paleontological data, you know that the clade was much more diverse several hundred years ago. And you can do the same for geographic ranges. You can also have misconceptions about the ecological importance of a species in an ecosystem, or in functional characteristics such as, say, body size distribution, as has been very elegantly shown, for example, for Chesapeake Bay oysters, where, again, if you use modern scientific data, you have a distribution of oysters that is relatively unchanged, but if you actually look deeper in time, it turns out that they grew to larger sizes and were actually more dense. The other interesting thing about paleontological data is that, in fact, even though we always care about biases in the fossil record, in reality, a very large proportion of species on our planet have some type of a skeleton that can be preserved and identified. And this is actually a subset of species that I got from IUCN Red List. So this is a Red List species of marine invertebrates, several common phyla, such as mollusks, arthropods, and corals, and islanders, I think. And you can see that regardless of the setting, whether it's marine, brackish, freshwater, or terrestrial, the black part of the bar is are species that are preservable, have high fossilization potential. And you can see that there are substantial subsets of the whole. And if you have a substantial subset, usually you have a lot of predictive power because you essentially can predict quite well, for example, distribution of species in a red list across different categories based solely on these fossilizable or preservable taxa. So there is enough data that is being preserved to give us a decent proxy. And I'm not going to spend a lot of time arguing about the quality of the fossil record. That would be a separate presentation. But I think we have increasing evidence that despite various challenges, we actually have data that are meaningful and they can provide meaningful information. So I will mostly focus on shells. That's my weakness, not only from a culinary perspective, but also in terms of research. And I will sort of argue that there are actually 7 reasons why shells can be a powerful tool for us. In fact, you can apply most of those reasons also to bones or teeth or tests of forearms, but I'm using shells of mollusks primarily. So the reasons are many. So the first and most obvious is that the youngest fossil record, the last centuries and millennia, is very well represented on the surface and subsurface on our planet. So almost anywhere you go, you can find fossils from the previous centuries and millennia. Technically, they are not really fossils because Holocene is often viewed as a time of subfossil record, but to me, they essentially function the same way. So here are various pictures from various settings: supratidal, shelf, aeolian deposits, river banks. Everywhere you can find shell material. Almost everywhere. And the other reason is that because we are dealing with very young fossil record, virtually all species that we encounter are species we know. So because they're still around today, and many of them are really well researched. And in many groups, such as mollusks, very often based just on skeleton, we can tell species, or at least genus. And with identifying species comes a plethora of information. You know, what species it is, where it lives today, what type of ecology it represents, and essentially, so this just by sheer fact of identifying it, we get a lot of data. Organisms also interact with other organisms, and in the process may acquire skeletal records, such as, for example, penetration mark from a casted snail that killed this particular poor echinoid, or a brachiopoid valve that was sequentially colonized by various encrusted organisms. And so you're not only learning about the history of the organism, but also other organisms that interacted with it. The more important reasons are advancement in instrumentation. So we have more and more tools that we can apply to biominerals, and we can extract information at higher resolution, more efficiently every year. This is improving. And so, for example, already 20 years ago, this is kind of a classic paleontological proof of concept study, we could use oxygen isotope profiles to track quite high resolution seasonal changes in temperature. And that's essentially for a living plant where temperature was recorded simultaneously with its growth. We can tie this to the growth rates to sort of estimate how temperature related to growth rates. And the issue is that these type of approaches can be translated into the young fossil record. More importantly, one of the key reasons why conservation paleobiology is a promising direction is the fact that geochronology for the very young fossil record is pretty good. We have tools such as radiocarbon. Previously, we used amino acid crystallization for dating of individual shells. Now radiocarbon dating advanced to a point where essentially it gives us much better quality data for the same price. And effectively, what that means is that we can date individual specimens, even relatively small ones, and based on that, we can develop geochronological framework. And with that, with several groups working on dating large number of specimens from single sites, we came to this amazing realization that what's on the surface, the superficial sedimentary on the seafloors and riverbanks, is actually time-rich. And typically, it's not very intuitive, but if you scoop a bunch of shells from a modern seafloor, or even from the shoreline, they will actually represent hundreds or thousands of years. This is a pretty extreme example from Brazilian shelf where we dated almost 300 shells, and you can see that we essentially had a continuous representation for 5,000 years. There are various reasons why this distribution is skewed in this direction, but that's less important for this part of the presentation. The point is that we have historical archives just on the surface that represent a lot of time. And now with the dating techniques, we can actually disentangle the time signal. And we also, of course, have advances in computation. So we have more and more sophisticated ways of analyzing the data. And now we have the advent of AI, which can also help process samples faster. And even with a simple laptop, you can do actually pretty interesting things to your data. So as our data becomes better and more complex, so do our analytical methods. So this is sort of my short sort of argument why conservation paleobiology is an interesting direction. And just to recap, young fossils are everywhere. By identifying species, we can get a lot of data without any extensive analysis. We can learn about biotic interactions. We have continuous improvement in methods, instrumentation methods, so we can acquire cheaper, faster, more precise, higher quality data. And we can date individual fossils, which allows us to really put everything in a chronological context. And by dating them, we've already realized that there's actually a wealth of time information in these assemblages. And of course, we can get all this wonderful data together and apply modern computational techniques to it. So, and then of course, potentially, where are we going with this? We're hoping that this can provide us with a long-term historical perspective about systems. We have diverse proxies that we can bring together to gain perhaps eco-environmental understanding of the past. And that may allow us to create some type of a baseline understanding for pre-human times. And by baseline, I do not mean a specific state. I rather mean a range of variability that naturally occurs in the ecosystems versus what we may observe in human disturbed systems. So it really sort of all fits to the long-term ecosystem dynamics. It can give us some historical perspective on ecosystem resilience. Even in certain cases, perhaps we can estimate the actual magnitude of anthropogenic changes against the natural system, or even estimate efficacy of restoration efforts. And the first example I'll show you, even though it's a bit naive, actually sort of highlights how you would go about doing this. This is not without challenges. One of my former postdocs came up with this really sort of interesting synthesis. This is not published yet, hopefully it will be soon, but where we came to a realization that while we are changing ecosystems, we're also changing the parameters that control how the fossil record forms. So for example, if you put an invasive species in a new environment and it's a heavy bioturbator, it will change mixing of sediment. This will affect what's preserved, what's the resolution of the record. And it turns out that there's a lot of human-influenced factors or processes that are changing, chemical, physical, biological, and they're actually affecting both the resolution and completeness of the fossil record that is forming. So we sort of think of this as somewhat analogous to Heisenberg's uncertainty principle, right? That the human-driven processes that alter nature also alter the record of those changes. And so that's, I'm of course overly optimistic about our abilities to do those things, but I think there are challenges that are on their own very interesting. So I'll go through these examples now. And again, I hope maybe I can do as many as 3, we'll see. And I'll start with a study that I've done, I have to confess, 30 years ago. This was from my dissertation times. And this is really sort of what the project that opened my eyes to the idea that actually paleontology can have some societal relevance. That, it's not all about the questions how many brontosauruses you need to kill a T-Rex. There are actually some things that are interesting that also may be relevant to us. So this project goes back to my early years when I was a graduate student and postdoc at the University of Arizona. And so this is a satellite image of the Colorado River Delta, which is virtually inactive because pretty much all water is being sequestered higher up. So So, you have various dams that have been constructed over the years. There is also municipal use of water, agriculture. So even at the Yuma, in Yuma, at the border with Mexico, there's almost no water nowadays. There's still some that is then used up in Mexico, and virtually nothing comes to the delta. Interestingly, this is one of those few cases when we have real instrumentation data going all the way back to 19th century. And that's actually in Yuma, at the border with Mexico, where we have measures of Colorado river flow, animal discharge, starting in 1875. And no offense to most of you, but trust Americans to come with a unit that no one can understand. Millions of acre feet. But I talk to some of you, and I understand that that's a lot of water. So this is a lot of water. It varies, as you may expect. If you have a better year in the Rockies in terms of snow precipitation, you get because most of that water comes from the meltdown from the Rockies. So you have higher and other in thinner years, but generally you see that this is pretty stable. Then we have a Hoover Dam constructed. It goes completely out of range here. So again, this is a natural what I refer as baseline. This is now, you can see outside the baseline. And then by the time Glen Canyon Dam is constructed, essentially nothing. In the 70s and 80s, we had few years when there was an unusually large amount of snow, and they put a fort to release some water to the Colorado Delta, and they sort of deemed this to be a restoration effort, right? So we bring in some water to the system, maybe that will help. And so that will become relevant in a moment. One of the things that I don't need to explain to people here is that if you shut down the river and all sediment supply in a macrotidal regime with very high tidal bulge, what happens is that sedimentation regime changes. And so all your fine sediment now is essentially carried away, and what stays is only large material, and the only large coarse material are shells. There's essentially no sand here or boulders, and those shells essentially accrete into these incipient ridges, and then through shoreline transport and tides, it essentially gets accreted to the supratidal zone, forming these amazing multi-generational ridges. And this is really, the picture doesn't give you the full justice of it. This is pure shell. So this is actually the surface of that picture. It's not a processed sample. This is what it looks like. But it's not very exciting to shell collectors because it's mostly just one species, the Molinia modesta, that I showed on one of the previous slides. And if you spend 5 or 6 days in the sun there, you start wondering, how many shells, right? You can't help it, and it's actually sort of my advisor, Carl Flessa from the University of Arizona, thinking about that very issue. And eventually we couldn't stand not knowing this anymore, so we came up with this kind of very naive back-of-the-envelope estimate. So we got high-quality satellite images, we got the total area of each ridges. This is underestimated because rocks are buried in the flanks. We estimated average beach ridge thickness by elevation, again underestimated because they're actually in the subsurface too. We then used shell density in beach ridges by both sampling. We estimated that, again underestimating because we only counted large deep shells. And with all these underestimations, we still came to a total number of 2 times 10 to the 12. And it's kind of funny when we were trying to publish this as a paper because in my sort of a second language, English as a second language ignorance, I forgot that trillion means different things in British English and American English. But just to be clear, that's what we are talking about. So it's 2 millions of millions. And that's a lot of shells, but that really doesn't tell you anything until you can put this in the chronological context. And that's what we did. So here you can see we have dated shells from these beach ridges, and essentially the bottom line here is that virtually all those shells came from the last 1,000 years. This decline here is probably an effect of sequential unroofing, starting with the youngest shells first. There may be also some decay-like loss of older material because it's exposed to various processes for longer. So we see this type of right skewness very often. This is, however, very unusual. In most of the distributions, the mode is the youngest age class. But of course, because of Hoover Dam, probably we know the answer. It's essentially the productivity of the system dropped dramatically. regardless, we can see that these shells that make beach ridges, 99% of them came from the last millennium. So now we know that at least 2 trillion shells were produced in the last millennium. We can use oxygen isotopes, look at the average sizes of these specimens to estimate what is the average age, ontogenetic age of the typical clam. And again, conservatively, we estimate about the 3-year turnover rate. So every clam occupied its quota of the intertidal habitat for 3 years. So 1,000 years, about 330 generations. That gives us about 6 times 10 to the 9th individuals. And the erosion occurs in the intertidal zone primarily, so there will be no sequestration area for all those shells. And again, everything here is very conservative because many shells are still in the tidal plasma wall of them being reworked. And we are pretty generous in terms of how far the erosion goes. It's probably less than that. But with all these underestimates, we got with 50 individuals per square meter. So we could claim in a paper that over the last 1,000 years, the average population density was about 50 individuals per square meter of the seafloor in the intertidal zone. And I thought, isn't that great to be a historical scientist? Prove me wrong, right? There's no other way of estimating this. And I was wrong because there are 2 Mexican paleontologists who discovered a fossilized tidal flap layer with bivalves in live position. They dated several specimens. They were all contemporaneous to one another within the radiocarbon era. And their estimate was something like 120 specimens per square meter. So well, this is actually great. Never expected anyone would test my paleontological results. That's unnerving. But actually, the results are pretty cool. So that made us a little more confident. If you actually look at density of living macrofauna, again, like restricted to mollusks, but in this case, we went over the top trying to make this number as high as possible. And we have a whole bunch of transects along the intertidal zone, and farther north, the tidal zone is wider, so the transects are longer, but they essentially go from supratidal zone to the lowermost intertidal. And you can see that we never get even close to 20 specimens per square, let alone 50. In fact, the average from that suggests that the density of adult shellfish was 20 times higher. And interestingly, all these surveys were done in 1999 to 2000, so it was after the restoration efforts, not before. And so this is one of the few cases where actually conservation paleobiology data were put to some use, not only from this study, but the leader of this project, my advisor, Frazer, continued working on this after this study really motivated it. And we've done a whole variety of studies of various sorts to sort of demonstrate diverse impacts of the river shutdown on the ecosystems of the intertidal zone. And essentially, this eventually influenced politics in the region to a point that the Minute 323 in 2017 actually allocated some water to nature. Very small amounts, not very satisfying, but essentially this minute is a treaty that deals with international water agreement between Mexico and US. And before, of course, it was just Mexico and US. There was no other stakeholders. Now we have also nature as a stakeholder, even though I think it's like measly 2% or something like that. Yeah, maybe to the river. So I mean, we hope that what we do with these historical perspectives can go into this direction, sort of help conservation biologists and environmental groups to sort of make their case and more strongly. You may recognize people here, so this is Tom Frazer and Sean Hyman in seahorses. And this is my second story, which builds mostly motivated by actual work of Sean. It's sort of an amazing work as a master's student, co-advised by Sean and myself. So we're now in a much more familiar territory. This is a whole series of sites. Actually, John's sites are only in steam hatching. We'll talk about those in a moment, but we have a whole series of sites where Tom and his colleagues have done research for a long time, monitoring water quality. They've also done other research on seagrass changes or changes in distribution of seagrass. And we sort of parasitized this, with his generous agreement, and we said, okay, why don't we unlike that comparisons of mollusks for these stations to see, whether life and death are matching. And there's quite a substantial literature that makes the case that while every region of the planet is impacted, the nature coast is still relatively unimpacted, comparing to many other regions that we still have a lot of healthy seagrass habitats. And we essentially test this hypothesis because if that's the case, then the living communities of mollusks associated with these seagrasses should be the same as long-term debt assemblages in the same area because nothing changed, or very little changed. And this is actually, again, we want to understand the temporal framework. We actually have more dates now, but the bottom line is that shells that we find on the surface in between the blades of seagrasses range over the last 2,000 years or so. And about half of them predate industrial revolution. So this is a long-term picture of the ecosystem here. It of course includes things that contributions from the youngest record, where we have human impacts that are significant, but the very large bulk of it is actually predating very substantial impacts. So it goes back to 19th century or earlier. And we can essentially look at kind of a regional scale. We can pull all the data we have on live mollusks and dead mollusks across all sites. And you can see that when we look at the top species in terms of rank abundance, there's actually a pretty good correspondence. So most of the live species that are in top 30 are also in the top 30 dead species. The rank orders are imperfect. There are a few species that are missing. Part of the reason why this is imperfect is because we have different proportional sampling of life and death in different estuaries, and that creates essentially unfair comparisons. It sort of forces mismatches. But we can do this at the site level, so we can go to every site that we have. I always come back here to switch the slides, even though I have this remote, so I'm clearly technologically challenged still. But what essentially we can do is to go to every site or every quadrant and compare life and death in terms of some simple metric of agreement. So we use sperm and rank correlation here, so it's rank abundance of life versus rank abundance of death. That value is 1, essentially the rank agreement is perfect. -1, of course, would be inverse. And you can see that these values hover around 0.5. They're never smaller than like 0.3 or so. And the mean is actually 0.53. And we think, well, that's probably not that impressive, the rank correlation of 0.53. But you have to realize that these are sparse data and uneven sampling or unbalanced sampling. Live samples tend to be much smaller. And so the sperm and rank is very heavily penalized for 0-1 combinations that occur just due to sampling. And this is a very simple simulation, or essentially a resampling model where we sample from pooled data. So we have a single rank abundance distribution, and we split this into the samples, into the actual sample sizes of our sampling scheme. And in that case, essentially, the correspondence is perfect. We sample the same system. And you can see that even if we do this, the mean sperm that we would expect is 0.72. So this is a sampling effect of sampling uneven samples with a lot of sparse data. So now, actually, 0.53 doesn't look that bad. It looks actually quite impressive. You could sort of almost correct it and say that, in fact, this is something like 0.8. And you can make a statistical case why this correction is reasonable. I mentioned, that some of it was motivated by Chan, and he's done essentially a study where he did live and dead comparisons for seagrasses and open stands. So we have two different habitats that form a mosaic here, and some of these actually are transitions where seagrasses were appearing and disappearing between subsequent surveys. When you look at living communities, so this is a simple NMDS ordination, where data points are samples. So of course, if they're very close to one another, the faunal composition is very similar. They're very far, they're very different. And not surprisingly, we know this from ecology, mollusks associated with seagrasses, green. And this is the similarity to USF colors is it's a much older slide. So, and the sand, of course, is here. You can see that there's actually more beta diversity in sand. It's much more variable in composition, but there is a very clear separation. That's not surprising. What's, however, quite fascinating is that when we look in the same ordination space at depth assemblage, time averaged over 2,000 years, they also separate, and they separate in the same direction. They're somewhat closer to the center of this plot, and actually, because of time averaging and mixing many generations, you would expect higher alpha diversity and suppression of beta diversity in these type of systems, and that will bring them closer together. But think about what this implies for stability of those patches of seagrass. If these seagrasses were shifting around all the time, so if my green side like this one, and my yellow side like this one, or these two sides, and we switching back and forth, so sometimes everything is sand, sometimes this is seagrass and this is sand, and vice versa. What we would producing then is a time averaged combination of habitats, and these things should really overlap. They shouldn't be differentiated. It's only under the scenario that you have relatively stable boundaries between these patches where you would expect to see any sort of discrimination between the two samples. Now, part of the reason why it's pushed to the center may be not just time averaging. Maybe it's not 100% of the time that this is seagrass. Maybe there are small fluctuations from time to time. But generally what this tells us is that what seagrass was probably mostly seagrass for most of the time, and vice versa. So this is really sort of a nice glimpse into a potential spatial stability of ecological landscape over time intervals that we cannot access using contemporary data. This is really bizarre. This is work of my former master's student, who is actually now a PhD student at USF in the Department of Geological Sciences. And she looked at drilling frequencies, so predation frequency that we can sort of calculate from dead shells, from depth assemblages, like all these clams that were killed by predators. This is restricted to a single species of clam. And we plotted, so again, remember, these drilling frequencies pertain to assemblage time average over 2,000 years. So some of these prey were killed 100 years ago, some 1,000 years ago. And what you actually see is that there is a remarkable correlation between modern estimates of total dissolved phosphorus, so some sort of proxy of productivity. If you plot phytoplankton concentration, you get very similar plot. So this comes from these stations that Tom and his colleagues ran, this data. And for the same stations, we see drilling frequencies. And you can see this amazing relation. It's almost like fake data. I made it up. I promise I didn't make it up. It's really, I couldn't believe when I saw it, and I still don't have an explanation, ecological or otherwise, why would that correlation be so strong. But it really does suggest that there is some permanency to this regional system. And maybe not surprisingly, because the productivity gradient is controlled by hydrogeology, and probably over the last 3,000 years, that was the primary driver and may have been stable. So, but don't ask me any questions about this because I don't understand why it's ecologically incomprehensible so far. Okay, so I think I have enough time to very briefly highlight one last project. This actually will move us to freshwater systems. This is related to Wacola River, and it involves Hurricane Michael, no relation to me whatsoever. And essentially, we are looking here at freshwater mollusks, freshwater assemblages, and we are trying to understand the life-death fidelity or correspondence between living and dead. And of course, we know from the outset that they will differ, right? Because we have invasive species that came in the invasive mussels, for example, that came in the last 30, 40 years. They probably will be absent or almost absent in the depth assemblages. So we sort of collecting data from these rivers, and specifically we're focusing on Wacola. And while we were collecting live and the data, the Hurricane Michael happened. So it's like, oh, that's really interesting. We can go back, and that's actually very insensitive. I mean, it's not interesting for people who live there, but it was a really sort of great opportunity for us to see what happened after the hurricane. We just collected live samples. And essentially, the upper part of Wacola is a state park, it's a protected area. and the lower part of Wakulla is open to boat traffic and other activities. And we also have data from Crystal River, but that's not relevant for this presentation. And so when you look at what happened, oh, and one more thing. It dated small terrestrial snails. There's new literature indicating that radiocarbon actually works for those, and we worked with experts who cross-tested this in terms of stability of dates, and they're pretty confident that the dates are meaningful. And what makes me confident that they are meaningful is that this is sort of a cluster of dates. So most of these shells are dating from maybe 12,000 BP to 7,000, 8,000 BP. Correspond to the Holocene transgression when all these things got reactivated, with the sea level rise. So that actually coincides with some data from lakes that my colleague Mark Turner assembled, and so it actually made sense, which makes me a little more confident. Regardless, again, we're dealing with a fossil record where our deck assemblages are actually representing mostly late Pleistocene and early Holocene. Some of it go back, back to as far as like 18,000 years ago, and then we have few young, like essentially one young specimen from the last 2,000 years. So this is definitely predating major industrial activities. It predates all invasive species for most of them. After our first survey, Hurricane Michael happened. It was a Category 4 hurricane, and essentially it created a storm surge that caused a complete reversal of the flow direction in Wakulla River all the way to the spring bed. And so that river essentially completely reversed its flow, and it had a bunch of various consequences. We collected the pre-hurricane data. This is sort of on the top here. So each of those corresponds to one of those stations. There was a particularly amorous alligator at this station, and we decided that our science was not worth it. So this is data for that. But you can see that each of the green shades is a native species, and different shades are different species. We have native species in the upper part of the river. We have also invasive species, and they increase towards sort of the lower part. Here we have actually invasive brackish gascrapode that is native, but it's actually not typically found in brackish conditions, in freshwater conditions. So we see that this system, without having any fossil data, we know it's out there. We know that these invasive species are new additions. We knew that this really shouldn't be there, and it's there. If we look after the Hurricane Michael, so that's 2 months after, we can see we killed him for some, I can't remember why, but there was a logistical reason we couldn't collect data from the head spring. But you can see that the invasive species are recovering or sort of dominating in many sites now. It's very commonly seen, apparently, if we recover from these type of disturbance events. This invasive species either encroached or already was there, we don't know, but we also have complete loss of native species at this station just below the park. And so there are some substantial changes. And 7 months after our species have improved, we have more native species here. So you can also get a sense that we're not something of, exhaustively. Probably the species were there, but maybe they were less common at that time. But we also see that the relative abundance of invasives dropped in the park, especially in the upper part of the protected area. Here we definitely see now encroachment of these brackish. This is one species of brackish snails on the right, and essentially it goes almost all the way to the bridge where the hard boundary is. But how big are those changes? Well, let's look at the fossils. And the fossils tell us, first of all, there's lots of species of native forms that existed in this river. At any given site, we have a lot of species. Grounded part of it may be time of origin. We have a lot of time in these deposits. But we can also see that they sort of spread through the entire river. Of course, we don't have any native species, but there's essentially much higher alpha diversity and much higher range for all species. And so from the perspective of what's there in the early Holocene when this river was active versus what's before and after hurricane, One could even argue that the hurricane impact was kind of trivial. I mean, everything that was serious about changing the system already happened before. Okay, so I think that I'm not going to torment you with one more case example. If I didn't make a case with 3 case studies, then the fourth is not going to help me. But thank you for your attention, and I'm happy to answer any questions.